Excellent. So, Chapter 1, a lot of this should be review for you all. If you've taken the prereqs for this course, you've probably seen a lot of the stuff we're going to cover in Chapter 1 already. So there are I've recorded the lecture for the material in Chapter 1. Again, if it's review and you already know it, you don't have to listen to me talking to you about it. But it's there for you in case you need a refresher or in case you haven't seen it before or for any reason. If you want to fall asleep to my voice, that's fine with me. So what are we going to talk about from this chapter? So we're going to talk about 3 main topics, the origin of life, as well as a review of functional groups, which you learned in organic chemistry. We're going to talk a little bit about cellular architecture, the structure of cells, and we're going to talk a little bit about thermodynamics, which is looking at the heat of reactions as they occur. So I'm not going to lecture to you on this material right now. That's for you to do on your own time, but just to kind of get you going, to maybe recall some of this information that you've seen before, I'm going to throw some questions at you and we can hopefully talk through them together. So does anyone have any idea what 4 elements are most prevalent in the human body? Yeah. Oxygen. Yeah, that's one. Carbon. Yep. Sorry, okay, my pen doesn't work. Hold on one second. There we go. Yeah, sorry, oxygen, carbon, and what? And hydrogen. Excellent. Yes, those are the 4 that are most prevalent. We do see a lot of the other ones, but they are not the 4 most prevalent. So, great job. Okay, what is the most commonly accepted theory for the origin of life on Earth? Yeah, go ahead. Yeah. Sorry, I didn't hear what you said.
I was going to say
hydrothermal vents. So that is a commonly accepted theory, but actually it is not the most commonly accepted theory. Does anybody? Yeah. Electricity in the atmosphere. Yeah, so yeah, I mean, the idea is that there were the chemical building blocks for life present in the atmosphere, and then electricity from lightning, as well as UV energy from the sun, help some of these molecules react with each other to start building the larger building blocks that are eventually became the biological macromolecules that we're familiar with today. Hydrothermal vents is another theory that is there's some evidence for. Some people are more in support of that one. And so in hydrothermal vents, you have heat, you have water, you have a lot of pressure. So it's not unreasonable to think that life could have originated in hydrothermal vents as well. You have sort of the building blocks for it. There's no evidence for sure that that is right or wrong. There apparently also is some amount of evidence that there is an extraterrestrial source. That is not a joke answer. I think in previous years a student did a project on the extraterrestrial theory and actually had some interesting information. If anybody wants to wow me with that this semester, I am open to that. All right, so let's talk about functional groups. Hopefully this is bringing back flashbacks from organic chemistry for everyone. So I just put up a couple of common pain relievers that you can mostly get over the counter that you may have seen before. So our first one on the upper left, we got Tylenol. So does anyone recognize that circled functional group I've got up there? Yeah, it's an amide, yes. So the amide has 2 parts. There's the amine part, the nitrogen part. Does anyone know what just this part is, the C double bonded to O? That's the carbonyl, yeah. So you'll see carbonyl groups as part of a lot of other functional groups, but when you see a carbonyl next to a nitrogen, that's an amide. And that, for anyone who doesn't remember, is spelled A-M-I-D-E. Hopefully you can see that. Yeah, all right. So on the bottom left, I have aspirin. Does anyone recognize the functional group I circled there? Yeah. Ester, excellent, yeah. So an ester also has that carbonyl group. but it's next to an oxygen. And that is an ester. Okay, in the middle on the top, we've got Advil. Does anybody recognize that circled functional group? Yeah. Carboxylic acid, yes. And that one also contains a carbonyl. And the other part of it, this OH, does anyone know what just the OH is called? Hydroxyl, yes, excellent. It's a hydroxyl. Sometimes it's also called an alcohol because hydroxyl groups define alcohols. Okay. Oh, and I forgot to ask, going back to our aspirin, so this whole thing is an ester. Just the oxygen bonded to 2 carbon. Does anyone know what that's called without the carbonyl there? Yeah, ether. Excellent. Yeah. So that is an ether. Cool. Oh, I already had one that was number 4 as well. Wow, I know my own slides. Okay. So in Aleve, I circled an ether for you. Great. So that brings us to Celebrex on the right. Does anyone know what the circled functional group is? We already sort of named it before as well. Yeah. Amine, yes, exactly. So anytime you see a nitrogen attached to pretty much almost anything else, it is an amine, some exceptions. So for example, this is an amide, it's an amine next to a carbonyl. So you can see the similarity in the naming because they are related molecules to each other. So that's great. And then in the lecture slides, there is a whole list of functional groups and which ones you're sort of expected to be familiar with for this course. And there are more practice questions on functional groups as well, if anyone didn't remember any of these. Okay, so prokaryotes and eukaryotes. So the first part, humans, that's us. Are we prokaryotes or eukaryotes? Yeah. Yes, we are eukaryotes. So can anyone tell me an example of a prokaryote? Yeah, bacteria, excellent. Okay, does anyone, I guess everyone just take a minute and write down 3 differences and similarities that you remember between prokaryotes and eukaryotes. Okay, so I think most of you are done writing. I don't know if everyone's done thinking, but we'll find out. So can anybody tell me one of the differences that they came up with? Yeah. That is true. Excellent. So prokaryotes, no nucleus. So eukaryotes do have a nucleus. Anybody else? Yeah. They're compartmentalized. Excellent. Yes. They're more compartmentalized. Can you tell me more specifically what makes the compartments in eukaryotes special? Yeah, so like that's like generally like what compartments are good for. Yeah.
Like membrane-bound organelles.
Exactly, yeah, they're surrounded by membranes. That's what makes eukaryotes special. Prokaryotes do have some differentiation, some compartmentalization, but they're not membrane-bound compartments. Okay. Yeah.
Prokaryotes have linear DNA or circular DNA, eukaryotes have linear.
Yeah, excellent. I'm just going to write usually because there are some unusual circumstances when you can have circular DNA in eukaryotes, but yes, that is almost all the time true. I think you'll have noticed by now that it's really hard to say always and never with biology and chemistry. It's a lot of times you'll say usually or most of the time. So be aware of exceptions. There's exceptions to almost every rule that we've come up with. Any other differences, similarities? Yeah. I'm sorry, ribosomes what? Oh, that's true. The ribosomes are slightly different in structure. I will say they both have ribosomes, but yeah, different. All right. Anybody else? Yeah. Yeah, so that is also one of those almost all the time things true. Yeah. There are some very rare circumstances where that can also happen in eukaryotes, but yes, that is true most of the time. Okay. Anybody else before I run out of room on the slide? Yeah. So yeah, that is true. I think almost all prokaryotes do have a cell wall. There are some bacteria actually don't have a cell wall. Also, can anyone think of a eukaryote that does have a cell wall? Yeah. Or, sorry, I wasn't really looking at either of you. You, sir. Yeah. Yeah. Plants. Yeah, it's an excellent example. But yeah, a lot of the eukaryotes that you think of commonly do not have a cell wall. For example, animals. So I'll write commonly no cell wall. or usually cell wall. Okay, so yeah, do you have another difference? Yeah.
Prokaryotes are always single-cell organisms. Eukaryotes can be single-cell organisms.
Yes, that is a good way to put it. Yeah, so prokaryotes are almost always unicellular. Eukaryotes are uni or multicellular. Okay. All right. I think we've all had enough of that. So let's move on. So we talked a little bit about how eukaryotes have those membrane-bound organelles. Can anybody name me one of their favorite or least favorite organelles, or an organelle they have no opinion about? Just any organelles. Yeah, ribosomes. So ribosomes are tricky. They're part of some organelles. They usually, when they're found by themselves, they're not a membrane-bound structure. Ribosomes themselves are made of protein and RNA. Can you tell me what organelle ribosomes are commonly part of? The rough ER. Yeah, exactly. Oops. The rough ER that contains ribosomes. And what does the rough ER do? What is its function? Or anyone? Yeah, excellent. Protein synthesis. And do you know the difference between proteins made in the rough ER compared to proteins made by free ribosomes within the cell? Yeah.
Are they usually exported out of the cell?
Yeah, exactly. So ribosomes that are on the rough ER make proteins that are going to eventually be either membrane integral within a membrane or that are secreted. Exactly right. So integral or secreted. Excellent. Any other organelles? Yeah.
I like the Golgi apparatus.
The Golgi? Yeah, why is the Golgi your favorite?
It's like the post office.
It is like the post office. How is it like the post office?
It distributes, it manages the distribution of everything that moves again.
Exactly, yeah. So it does some further modifications of proteins and it sends them to the proper location. Anybody else? Favorite or least favorite organelles? Yeah.
The smooth they are.
Uh-huh.
For detoxification and steroid and hormone function.
Yeah, excellent. So what we'll mostly be talking about in terms of the function of the smoothie, it has a lot of functions like you mentioned. What I want you to remember about it is that that's where lipid synthesis is happening. But also a number of others. So what else we got? Yeah. Uh-huh. Excellent. Yep. All right. Any other ones? Yeah. Lysosomes, great. What are lysosomes for? Yeah, exactly. It's sort of for degradation of waste products. There is a big one that I think you all know. Yeah? The nucleus, yes. Yeah, exactly. It's just some people call it like the information center. It's for storage of your genetic information. Mm-hmm. Okay. That is most of the big ones. Oh, there's, yeah, I think that's it. I was also going to say that some people, or another important part of a cell to know is the cell membrane, but that is not an organelle. The cell membrane surrounds the whole cell. Okay, so let's move on. So we are going to talk a little bit in this course about evolution and how that affects living organisms. So something that we've all been kind of watching over the past few years is the virus SARS-CoV-2. So can anyone tell me a little bit how it's evolved over the last few years since it came into our lives? Yeah, less virulent. Yeah.
It has a very rapid mutation between new strains that come out.
Yeah, I mean, so that's sort of like what I was getting at exactly. So how do these functional changes happen? The changes in virulence happen because of mutations, like you're saying. So as the virus replicates, there are just random errors that occur every time it copies its genetic code, which in the case of SARS-CoV-2 is actually RNA. But as those changes happen, the proteins that are coded for by that RNA might make small changes, and that might make the virus, for example, make you less sick. It might make it more or less contagious. It might, make it do different things to hide from your immune system or make it more visible from your immune system. Yeah, does anyone sort of know where, like in what how to put this, like how where the changes might have happened? Like are the changes happening in like really healthy people who recover quickly? Are they happening in really sick people? Like what does the virus need after it makes the changes to be able to pass those changes on?
So it needs to evolve in healthy people, because if it kills its host, it's not going to get really a chance to evolve and reproduce itself, right?
Yeah, that's a really good point. Exactly. So viruses, they always need a host to be able to replicate and spread to other hosts. One host that a lot of viruses, especially SARS-CoV-2, really like are people who have compromised immune systems. So these people, when they get infected, their immune system tries to fight it off, but the virus can sort of learn from it because their immune system is hobbled in a way. It's not as effective as a healthy person's immune system. And so the virus, the virus can get resistant to some of these attacks from the immune system, and those more resistant viruses can be selected for. So multiple changes can happen as the virus sort of thrives within an immunocompromised person. Okay, so now we're going to switch gears a little bit and talk a little bit about a review of thermodynamics. So you have probably learned these types of things before. We'll see if anybody remembers what is enthalpy. Mm-hmm. Sort of, yeah. I guess maybe a better question is, yeah, like what does the change in enthalpy represent? So it's as a reaction happens, when enthalpy changes, what's happening? Yeah. Yeah, exactly. So enthalpy represents the heat being lost or gained by a reaction. Does anybody know what symbol is used to represent enthalpy change? Yeah. Delta H, exactly. Does anybody remember for an exothermic reaction, is delta H positive or negative? Yeah, negative. So it's negative. And what kind of reaction has a positive enthalpy change? Endothermic, excellent. Yeah. Okay. So I forgot that that was already in my slides. So we already said that when heat is lost, delta H is negative. When heat is gained, delta H is positive. So spontaneous reactions, do they tend to be endothermic, exothermic, or isothermic? Yeah, exothermic, yeah. Not always, but when a reaction is exothermic, it's more likely to be spontaneous. Great. So let's talk about entropy. Anybody know what entropy is? Yeah. Exactly, yeah. So entropy is a measure of disorder or randomness. What symbol do we use to represent entropy change? Yeah, delta S, exactly. On the right over here, there are two figures, A and B. Which one has higher entropy? Which one looks more disordered or random? Yeah, B, yes. So B, because these molecules are distributed in all over the place, they are sort of more random. In part A, they're all enclosed in one side, making this more orderly. Okay, so spontaneous reactions, do they tend to increase or decrease the entropy of the system? Yeah. Yes, so when you increase entropy, it is more likely for your reaction to be spontaneous. Not always, but it helps. Okay, so that kind of brings us to the Gibbs free energy. Does anybody remember the Gibbs-Helmholtz equation? Yeah. Yeah, excellent. So we have, I think I have that up there. Yes. Oh, sorry. So I'll just write that in for now. So delta G equals delta H minus T delta S. So if delta G is positive, what does that tell you about the reaction? Is it spontaneous, non-spontaneous? Yeah. Yes, non-spontaneous. If delta G is negative, what kind of reaction do we have? It would be spontaneous, thank you. And if delta G is 0, what is happening? Equilibrium, exactly. The reaction is not going anywhere. Great. So now we can talk a little bit more about that Gibbs-Helmholtz equation. So if delta H is negative and delta S is positive, what does that tell you about the reaction? Is it always spontaneous? Is it never spontaneous? Yeah.
That's kind of a question.
Sure.
On the previous slide.
Okay.
If delta G is at equilibrium,
Yeah.
does that mean it's also non-spontaneous?
No, because it would have to be positive to be non-spontaneous. So You can think of a non-spontaneous reaction as being spontaneous in the reverse direction. A reaction that's at equilibrium is not spontaneous in either direction.
Okay.
There's nothing pushing it to either direction. So if delta H is negative and delta S is positive, what does that tell us about delta G? Yeah, always spontaneous. That is true because delta G will always be negative. Okay, so if delta H is positive and delta S is negative, then what does that tell us about delta G and the reaction? Yeah. So if delta H is positive and delta S is negative, delta G is going to be always positive, right? Unless I'm doing math wrong. So if delta G is always positive, right, then what does that tell us about our reaction? It's never spontaneous. Excellent. Yeah. So now we get into these other two. So if delta H is negative and delta S is also negative, then what do we have? Yeah.
Spontaneous at low time and then non-spontaneous at high time.
Yeah. So when t is small, delta G is dragged to be a more negative number. When t is large, delta G can be dragged into a positive number. So spontaneous at low temperatures. So then what does that tell us about when delta H and delta S are both positive? Want to go for it? Thank you. Yeah, for the... So when t is low, delta G gets dragged to be negative, so it's spontaneous at, or I said that backwards. When t is large, delta G is negative. Okay, so all of that is in your textbook, in the lectures that are recorded. It's basically math. I'm sorry, there's math, if you don't like math. That is actually all the review I have for you today. So I know it's a little bit before the class is over, but if I will let you go for now. I recommend that you watch the lecture slides, try out those practice questions that are posted. We'll go over chapter 1 on Thursday. There's some chance we'll get through chapter 1 and start chapter 2, and I will post the chapter 2 slides and lectures hopefully in the next couple of days so you can get a head start on those. And please try out Quiz Zero. And if you have any questions, you can talk to me right now, or we can go chat in my office if you have a longer question. Thank you very much.